You probably didn't realize it, but you started learning math long before your first math class. You learned to count even before starting school. Counting is the first step in learning math. Once you had a foundation in counting, you could learn addition and subtraction. Once you mastered adding and subtracting, you moved on to multiplication and division. You couldn't learn multiplication before you mastered addition, because you didn't have the proper foundation to build on. The process of building on previous understanding continued with each new math concept you learned. As you mastered one concept, it became part of the foundation for attacking the next concept. In this way, learning math is a gradual process. Like learning math, the accumulation of biological knowledge by researchers is also a gradual process. Biologists use information from previous experiments as a foundation for future discoveries. The steps in the discovery that DNA is the genetic material and the experiments used to determine the three-dimensional structure of DNA are perfect examples of the process of biological discovery. Biologists learned first that structures in the cell called chromosomes are the mediators for inherited traits. Once biologists understood the importance of chromosomes, they were able to determine that the material in chromosomes that holds the hereditary information is deoxyribonucleic acid, or DNA. The next step was to discover DNA's structure. In this picture presentation, we'll look at the series of experiments that led to the discovery that an organism's genetic information is found in its DNA. Then we'll look at some structural details of DNA. Finally, we'll discuss the implications of DNA structure on the mechanism of DNA replication. DNA structure makes the basic method of DNA replication simple and accurate. It's a good example of the relationship between structure and function in biology. Biological discovery is a process of building on earlier achievements. Several important steps led to the discovery that DNA holds the hereditary information. Let's look at how biologists first determined that chromosomes are the mediators of inherited traits. In the late 1800s, Gregor Mendel, an Austrian monk doing experiments with pea plants, discovered that specific traits are determined by inherited factors, later called genes. Mendel's work went largely unnoticed until the early 1900s, when American biologist Walter Sutton and German embryologist Theodore Boveri independently noticed that the behavior of chromosomes in egg and sperm development exactly matched Mendel's theory of inherited genes. This led them to believe that Mendel's genes are located on chromosomes. They proposed the chromosome theory of inheritance, which stated that chromosomes carry genes, the hereditary information. The chromosomal theory of inheritance was further supported when Thomas Morgan was able to figure out that the gene that gave fruit flies red eyes was located on a specific chromosome. The next groundbreaking experiment in the discovery of DNA as the genetic material was the first bacterial transformation, performed by British medical officer Frederick Griffith in 1928. Bacterial transformation is the process by which bacteria are changed by the uptake of foreign DNA. But at the time of Griffith's experiment, nothing was known about bacterial transformation. Griffith was studying the bacterium Streptococcus pneumoniae, which causes pneumonia in mammals. Griffith had two strains or versions of the bacteria, one that was smooth because of its polysaccharide coat and one that was rough because it lacked a polysaccharide coat. Griffith found that the smooth cells were able to kill injected mice, while the rough cells could not. When he used heat to kill the smooth cells, they were no longer able to kill the mice. However, when heat-killed smooth cells were mixed with the non-lethal live rough cells, the injected mice died. And live smooth bacteria could be recovered from the dead mice. Somehow, the rough cells had acquired the ability to make the polysaccharide coat and were now able to kill the mice. At the time, Griffith didn't know what made the rough cells become smooth cells. However, his experiment gave other scientists a starting point to look for the nature of the genetic material responsible for the transformation. In 1944, American bacteriologists Oswald Avery and Colin MacLeod used the data from Griffith's experiments to determine what the transforming agent was. They purified various substances from bacterial cells and tried to transform rough bacterial cells with each of the substances individually. Avery and MacLeod found that only DNA was able to transform the bacteria and make the bacteria deadly to the injected mice. The rough cells picked up the smooth cell DNA and began making the polysaccharide coat. The ability to make the polysaccharide coat was then passed from generation to generation. This observation established that DNA is the genetic material. In 1952, American biologists Alfred Hershey and Martha Chase devised an experiment to determine if the genetic material in a bacteriophage is protein or DNA. A bacteriophage, or just phage for short, is a virus that infects bacteria. Viruses are much simpler than cells and are composed of DNA enclosed in a protein coat. Scientists hypothesized that the genetic material of the bacteriophage must be injected into a bacterial cell during an infection. Hershey and Chase used radioactive isotopes to keep track of the locations of the protein coat and the DNA during an infection. A radioactive isotope is an atomic form of a chemical element that is unstable. To reach a more stable state, the isotope gives off a detectable particle. Hershey and Chase incorporated radioactive isotopes into the bacteriophage. Proteins contain sulfur, but little phosphorus, so they used radioactive sulfur to indicate the location of the protein. DNA contains phosphorus, but not sulfur, so they used radioactive phosphorus to indicate the location of the DNA. Hershey and Chase incorporated the radioactive isotopes separately into 2 samples of bacteriophage and then used the radioactive bacteriophage to infect bacteria. They placed the infected bacteria and bacteriophage mixture in a blender and agitated it. Only bacteriophage material that was injected into the bacteria would remain in the cells. All other bacteriophage material became loose in the medium. Hershey and Chase separated the bacterial cells from the medium in which they were growing. Bacteriophages inject only their genetic material into bacteria, so bacteriophage DNA, but not protein, was found in the cells. Bacteriophage DNA, marked with radioactive phosphorus, entered the bacterial cells. The protein coat, marked with radioactive sulfur, remained outside the bacteria and was found in the medium. This experiment further supported the idea that DNA is the genetic material. With the discovery that DNA is the genetic material, the race to uncover the three-dimensional structure of DNA began. The arrangement of the bonds in a DNA polymer was already known. Each monomer unit, or nucleotide, of the polynucleotide chain is made up of a phosphate group, the sugar deoxyribose, and a nitrogenous base. The phosphate groups and deoxyribose sugars form the backbone of the chain. There are 4 nitrogenous bases, guanine, cytosine, adenine, and thymine, abbreviated G, C, A, and T. The Austrian biochemist Erwin Chargaff found that the amounts of adenine and thymine found in DNA were always about equal to each other, and the amounts of guanine and cytosine were always about equal to each other. These observations are known as Chargaff's rules. At the time, nobody understood why Chargaff's rules were true. Determining the three-dimensional structure of a DNA molecule required data from several groups. In the early 1950s, while working in London, Rosalind Franklin produced photographs of the X-ray diffraction patterns of DNA fibers. In 1953, from Franklin's X-ray photographs, an American James Watson and an Englishman Francis Crick determined that DNA is a spiraling coil called a helix. They were also able to tell that the helix was about 2 nanometers wide. This fit with DNA being a helix of two polynucleotide chains, commonly called a DNA double helix. In a DNA double helix, each turn of the coil is 3.4 nanometers long, and each nucleotide is 0.34 nanometers long. This means that there are 10 nucleotides per turn. The width of the double helix also helps scientists understand Chargaff's rules. Adenine and guanine have 2 rings. They are called purines. Thymine and cytosine have only 1 ring and are called pyrimidines. To fit the width of the double helix, a purine on one strand must pair with a pyrimidine on the other strand. Chargaff's rules state that there is the same amount of adenine as thymine, so adenine pairs with thymine. Similarly, guanine pairs with cytosine. The purine and pyrimidine are held together by hydrogen bonds. Another interesting aspect of DNA structure is the orientation of the 2 strands in the double helix. The strands run in opposite directions. This is called an antiparallel orientation. The carbon atoms in the deoxyribose sugar are carbon 1 to carbon 5. A phosphate group is bonded to the number 5 carbon, known as the 5' carbon, and another phosphate group is bonded to the number 3 carbon, the 3' carbon. For simplicity, we'll place one strand with the 5' carbons all oriented to the top of the screen and the 3' carbons to the bottom of the screen. Since the other strand is antiparallel, all of the 5' carbons will face the bottom of the screen and the 3' carbons will face the top of the screen. As we read the nucleotides down the screen, the strand on the left is 5' to 3', and the strand on the right is 3' to 5'. This is a piece of double-stranded DNA. When we break the hydrogen bonds holding the strands together, we end up with 2 single-stranded DNA molecules. We can use the single strand of DNA to figure out the sequence of nucleotides on the second strand. To determine the sequence of nucleotides on the second strand, remember that adenine always pairs with thymine and guanine always pairs with cytosine. To find the sequence on the second strand, the original strand is used as a guide or template. Both new pieces of double-stranded DNA are identical to the original double-stranded DNA. Each time a new discovery is made, scientists think about its implications. What are the implications of the discovery of DNA structure? Let's think about the role of DNA. DNA holds the information needed for an organism's growth and survival. Multicellular organisms start as a single cell. The single cell divides, and the resulting cells continue dividing as the organism grows. Changes in its DNA are generally not beneficial for an organism's health. Therefore, each cell needs to have the exact same DNA as the original cell. Every time a cell divides, it must make an exact copy of the DNA to pass on to the daughter cell. Once a change in the DNA occurs, it will be passed on to all daughter cells. For the health of an organism, DNA replication must be very accurate. The structure of DNA provides a perfect mechanism for ensuring that DNA replication is accurate. Because adenine always pairs with thymine and guanine always pairs with cytosine, a single strand of DNA can serve as the template for copying the other strand. One double-stranded DNA molecule can be used to produce 2 identical molecules of double-stranded DNA by copying each individual strand independently. Learning is a gradual process. Each new concept builds on previous information. As we master the new concept, it becomes part of the foundation for learning the next one. This is true for learning concepts others have already figured out, like basic math, and also when investigating topics that no one yet understands. We looked at the steps in the discovery of DNA as the genetic material. Mendel, Sutton, Boveri, and Morgan provided the foundation with their ideas about the presence of genes on chromosomes. Griffith performed the first bacterial transformation experiment, which gave Avery and MacLeod a starting point for their discovery that DNA is the genetic material. Hershey and Chase confirmed this with their experiments using radioactive bacteriophage. Once biologists were convinced that DNA is the genetic material, the next step was to figure out the structure of the DNA molecule. With help from data collected by Franklin, Watson and Crick determined the double helical structure of DNA. Using the width of the double helix and Chargaff's rules, Watson and Crick found that the nucleotides pair in a very specific way. Adenine pairs with thymine, and guanine pairs with cytosine. The two strands of DNA are antiparallel, meaning they run in opposite directions. Bases on opposite strands are held together by hydrogen bonds. The structure of the DNA molecule is a good example of the structure-function relationship. Accurate DNA replication is very important for an organism's health. Changes in an organism's DNA are not usually beneficial, and once a change occurs in the DNA, it's passed to all daughter cells. The structure of DNA provides a very accurate mechanism for DNA replication. Because adenine always pairs with thymine and guanine always pairs with cytosine, a single strand of DNA can serve as the template for the other DNA strand. One double-stranded DNA molecule can be used to produce two identical molecules of double-stranded DNA by copying each individual strand independently. The process we just saw in the discovery of the role and structure of DNA is typical of biological discovery. Biologists use information collected previously to answer new questions and solve new problems. Each discovery is a stepping stone to the next discovery.